Hello, I'm Laura Shepherd from the Faculty of Dentistry, Oral and Craniofacial Sciences at King's College London. With me today is Guy Carpenter, who is a Professor of Oral Biology. Many thanks, Guy, for taking part in the Spotlight on COVID video series. To start with, if you could tell us about your work at King's College London, just in general.
Sure, I've been at King's College for about 25 years now, did my PhD here, and that started my interest on saliva. So I do research on just about every application of saliva you can think of. So it started off looking at some of the proteins in saliva, but I've been more recently interested in taste and how it affects the things that we eat. But I have a wide range of interests, and also looking at some of the microbiology and obviously more recently some of the virology of saliva. Yeah,
that's great. So it'd be good to hear about your research that you're doing. You're just starting a new project which is looking into COVID-19.
Yeah, so we got some funding recently, and that's to look at using saliva samples to try and help manage or understand this COVID infection pandemic that's occurring. And we already know that the swab test that people are using is picking up viral RNA, so it's trying to detect live virus. But there's a big need, I think, to understand who has been infected and whether they have any immunity. So what we don't know about COVID yet really is how well the body responds to that virus and whether it makes any antibodies. Because if it doesn't make any antibodies, that means we're more susceptible to further infections by the same virus. So this project is really to try and look at how the body responds to that viral infection. And one of the ways that it responds is to make antibodies. And the main antibody that you have in saliva and other mucosal sites, like your lungs and your throat, etc., is an antibody called secretory IgA. And so the project is really trying to identify the part of the virus that the secretory IgA wants to react against. So we make lots of antibodies. They line these mucosal surfaces like the mouth, and if they're there, then they can detect and interact with the virus and prevent infection. So that's why it's important to try and measure those antibodies. The tricky thing is, though, understanding what those which part of the virus those antibodies are directed against. And so that's what this project is going to look at.
That's great. So why did you decide to look into this topic? What kind of potential does it have?
Yeah, so I've been interested in saliva, and so a salivary test has always been, there's been a great hope that saliva would be a great diagnostic medium for a whole variety of things. And, you know, so 10, 20 years ago, for example, HIV testing was another example where saliva held a lot of promise for diagnostic testing. And to a large extent, it is quite well used for HIV testing, because obviously with blood, it's more infectious. HIV is more infectious. So obviously it's a sort of area of interest to see whether we could make not only a diagnostic test, because I think in some ways that's already present, is to really look at this question of immunity and whether people will be, firstly, if they have the virus, whether they build any immunity to it. Secondly, if we develop a vaccine, whether that vaccine will also induce these kind of antibodies. So I think that's the sort of general area in which it may help.
That's great. When do you expect to see results? How will you go about conducting this research?
Yeah, so we're starting off just by trying to collect saliva samples from people who have had the virus infection. We're collecting about 4 weeks after the onset of symptoms, and the reason for that is that the viral load will have decreased significantly, so that the samples are less infectious, so it makes it less dangerous for us to work on the samples. Because principally what we're interested in is the antibodies, and those come after the infection usually. I mean, often they start being developed during the infection, but if they're going to provide immunity, they need to be there, you know, months, years after the infection. So what we're going to do is we're going to invite people to provide a saliva sample, but we're going to send a saliva collection kit to their home to collect the saliva sample. And in that kit, there are detergents that help to break down the virus so that the saliva sample is no longer infectious when they send it back in. Because, of course, one of the great problems is working on this area is that we're not sure how infectious saliva is. There's been reports that there is live virus within the saliva. And so we have to take the utmost care that any samples that we collect from people who have been previously infected are no longer infectious. So that's a sort of handicap that we sort of have to be aware of.
Great. And what's kind of the future for this? How will this help down the line once you've got all the results?
Yeah, I mean, I think it will allow, you know, if you can test people for their immunity to the virus or levels of neutralizing antibodies, these secretory IgA antibodies, it might help to allow people to come out of self-isolation, for example, to say that they're non-infectious to other people, or may provide reassurance that that they can re-enter a workplace, for example, without infecting other people. We're still not sure whether that can occur at the moment. Like I say, it can also help maybe with analysing how successful vaccine studies, although typically what they would do there is to take a serum sample. But obviously we're not going to do that on everybody because it's quite invasive and time consuming to take blood samples from everybody. So saliva has this, you know, benefit of being very easy to collect, much easier than these nasal oral swabs, which seem to be quite difficult. So I think that's the sort of obvious applications, yeah.
That's fantastic. When do you expect to have any kind of results on this?
Yeah, so I think it's hard to tell, isn't it? But we sort of we would guess in the sort of 3 to 6 months, maybe out to a year, kind of. It depends how quickly we strike gold, I suppose. So we're using a structural biology type approach to this. So what we've been doing is to try and help identify those targets. We've been looking at the proteins that we think are important, and the spike protein seems to be the important one. And we've been comparing the structure of that with a previous SARS virus, and then looking for any differences, and also at other studies where they've found neutralizing antibodies. So typically those neutralizing antibodies have been IgG, serum antibodies. But so we're going to specifically look at secretory IgA. But yes, we're using a structural biology approach to try and identify those targets, because it can be, you know, the targets can be very small. It can be a needle in a haystack kind of thing. But by using this structural biology approach, we're hoping to sort of speed up the whole process. But it may be that actually it's very difficult to find, and so therefore it could take a year into years, or
yeah,
who knows.
Well, fingers crossed that it's gets done pretty quickly and we get some results that can help everyone.
Sure.
Who's supporting the research apart from King's College London? Are any other funding bodies helping out?
Yeah, so we got a funded grant from the Biomedical Research Consortium here at King's. So they put out a call a little while ago, and so we got a little grant from them to just sort of prime it, I suppose, and fund the next stage. But obviously we'll be looking for larger funding soon.
That's brilliant. All right. Thank you so much for your time today, Professor Carpenter. We look forward to seeing some results soon.
Okay, thank you very much.